Okay, so in photosynthesis, we always target with the light-dependent reaction. And always remember, light-dependent reaction is going to produce two kinds of molecules that they are required by the Kelvin cycle, which are ATP and reduced NADP. Always remember, these two items or these two compounds are required by the Kelvin cycle, ATP and reduced NADP. So to produce ATP, or we say the photophosphorylation of ADP to ATP can go through by these two processes, either cyclic or non-cyclic, depending on the patterns of the electron flow in one or both type of photosystem. If we look at this, if cyclic, if the ATP formation through cyclic, then only involve photosystem 1. Are you clear? If you want to form, if the plant want to form the ATP, okay, through cyclic photophosphorylations, then they only need photosystem 1. But if the plants want to use non-cyclic process to produce ATP, then they have to go through photosystem 1 and photosystem 2. Both photosystems are involved. Now, how about reduced NADP? Right now, to produce reduced NADP, only one must have both photosystem 1 and photosystem 2 through non-cyclic. Are you clear? Right now, reduced NADP must be non-cyclic because cyclic won't produce. Cyclic photophosphorylation, the process won't produce reduced NADP. There is a lot of debate about this, okay? Because when you look at this process, we realize that if the plant would use non-cyclic photophosphorylation, they will be able to produce ATP and reduce NADP together, right? So I say that's right. ATP can be cyclic, can be non-cyclic, reduced NADP must be non-cyclic. So it means that the plant use non-cyclic, basically they can produce ATP and reduced NADP together. So there's a debate why plants still need to have the cyclic. Okay, and in fact, majority of the process is non-cyclic. Cyclic photophosphorylation actually is a very minority. Okay, depends on the different type of the plant species and also the plant tissues. Okay, so let's start with the non-cyclic photophosphorylation first. So this is the pathway. Let me go through the pathway, what you need to know and how you write it. Okay. So now, in terms of electron flow, always remember, in terms of electron flow, on paper, the flow will be start from the water molecule, go into the photosystem 2, pass through the electron transport chain, enter into photosystem 1, and then go through the electron transport chain again, and finally received by the NADP to produce reduced NADP. That is the flow. Are you clear? Now this flow, don't look at it, oh, it seems the electron jump up and down, because some books also use the term called Z-scheme of electron flow. But in fact, you won't see the electron actually jump up and down like this. Are you clear? Because this up and down refer to the energy of the electron. Because when the photosystem means that the primary pigment receives the electron, the energy, the light energy, the electron actually is photo-excited. Photo-excitation of electron actually takes place. So this electron has high energy. So this high energy electron pass through a series of electron carrier and finally received by photosystem 1. But in order to explain it better, I always start with photosystem 1. So you can see process number 1, process number 2, process number 3, and process number 4. So what actually happened here is, look at this, guys. I always explain here photosystem 1, where our reaction center P700. So what actually happened is this P700. Basically, these P700s receive the light energy. Or light energy can be received by the accessory pigment. This energy passes to the P700 reaction center. Once they gather sufficient energy, photo excitation of electron actually takes place. Are you clear? The electron actually coming out from the magnesium. So magnesium now become Mg2+. The electron actually move on. Don't worry about the form here, right? Don't worry. So this electron will be captured by the electron transport chain. Don't worry about paradoxine. So when this process takes place, energy release. Okay, and finally accepted by who? Accepted by this NADP. NADP is similar to the NAD, but they have extra phosphate group. Okay, NADP, clear, similar to NAD that we have learned in energy respiration. Slightly different, they have extra one phosphate group. So that's why we call them NADP. So this NADP, okay, together with the hydrogen ions, okay, so together with the electron, combined together become reduced NADP. Write down, this one is reduced NADP. The enzyme involved here is the NADP reductase. I will draw it later, so write down first. Okay? Yeah, so again, receive the photon. Photon means light energy. Receive light energy, pass the light energy to the reaction center. Photo excitation of the electron. Electron pass through electron transport chains received by the NADP. NADP together with the proton and electrons combined together become reduced NADP. And this process catalyzed by the enzyme NADP reductase. First part. But the thing here is, my reaction center don't have electron already. Basically, kick out 2 electrons, no more electron. We need to replace the electron. So at the same time, always remember that light energy won't strike on one photosystem. At the same time, it strike both. So electron flow actually this way. Can you see that? Are you clear? So it means that on the same time, at the same time, what happened here? Photosystem 2. At the same time, photosystem 2. Sorry for those online, I forgot. Okay. Now, photosystem 2 in this case. So for photosystem 2 also receive the photon. What happened? The light energy, this energy will pass to the reaction center P680. Again, photo excitation take place. And this electron will pass through this electron transport chain, and where chemiosmosis take place. Still remember chemiosmosis? Still remember chemiosmosis in the mitochondria. So this chemiosmosis take place, allow the formations of ATP. Can you see that? So these electrons from the photosystem 2 now flow across this electron transport chain and finally received by photosystem 1. So it means that I replaced already. Because initially photosystem 1, the electron received by the NADP, right? Not? So lack of electron. So from photosystem 2, electron now come back to photosystem 1. But same problem faced by the photosystem 2 because they donated the electron. So once they donated the electrons, means that they lack of electron. Someone have to donate the electron to photosystem 2. So who donate the electrons now? You can see the water molecule. So the enzyme, water splitting enzyme, helps to catalyze these reactions. So in these reactions, you can see that water molecule is broken down to hydrogen ions and also the electrons, where oxygen is released as the byproduct. And this process takes place, write down guys, this blue color take place inside the thylakoid space. The rest take place at the thylakoid membrane. Clear so far? Any question? Okay, to summarize this non-cyclic photophosphorylations. So first thing, we're going to look at this photosystem 1 first. Okay, we start with the photosystem 1. Okay, So what happened here is photosystem 1, at the center here, we have the reaction center. This reaction center is termed as P700. Okay, photosystem 1, P700. So what actually happened here is the accessory pigment received the photon and pass this light energy, pass this energy to the reaction center. Are you clear? So once it pass this energy to the reaction center, so I write here, this is the first process. So what is the first process? So first process, okay, so pigments, okay, or photosynthetic pigment, or chlorophyll, anything lah, okay, photosynthetic pigments absorb the light energy and pass this energy to the primary pigment at reaction center, T700. So what is the effect here? Number 2, then you have the photo excitations of electrons. So it means that now electrons have higher energy. Doesn't mean that electron actually jump up this electron. Are you clear? So this electron will pass through a series of electron carrier. So these electrons now pass through, say, from one electron carrier to another electron carrier. So this part we call as ETC. Okay, so this is process number 2. So until this electron received by who? These electrons now, together with the hydrogen ion, together with the NADP, form reduced NADP. So the enzyme involved here is NADP reductase. Okay, so this is the third process. And number 4. So it's number 3. So electrons are, is transferred along electron transport chain, and then these electrons together with the proton, okay, they form the hydrogen atom. So this hydrogen atom accepted by the NADP to form reduced NADP. So the enzyme that catalyzes these reactions is NADP reductase. So what is the problem here? The problem here now, reaction center or primary, the reaction center in PS1 now lacks of electron. Okay? Clear? Clear? So this process actually produced the first item needed for the Kelvin cycle. Okay? First item needed for Kelvin cycle. So now we have to replace. So how to replace? Then we have to ask the help from photosystem 2. So now come to photosystem 2. So photosystem 2 also have the reaction center. So this reaction center is termed as P680. Are you clear? So again, they have the accessory pigments. So they also receive the light energy and pass this energy to the reaction center, and photo excitations of the electron take place again. Okay? And these electrons now pass through a series of electron carrier. Okay, pass through electron carrier. And finally, accepted by the P700. Are you clear? These electrons. But during this process here, guys, this ETC, the transport change. So during this process, you can see that chemiosmosis takes place. So what is the main events? ADP together with the inorganic phosphate group become ATP. Are you clear? Can you see that? Here, so it produces the second items required for the Kelvin cycle. So write down number 4, this one number 5, number 6, number 7. Okay, so number 5, event number 5. So again, photosynthetic pigment absorb the light energy and pass this energy to the reaction center, which is P680. So what is the event take place? So photo excitation of electron. Are you clear? So next, this electron is transferred. Sorry. Along the ETC, and this very important process here, chemiosmosis take place. I will explain in detail where ATP is produced. Are you clear? And finally, these electrons accepted by P700 at photosystem 1. Clear? Clear? Now what's the problem? Photosystem 2 now lack of electron. So we have to carry out photolysis of water. Of H2O. So basically H2O, get the H plus electron plus O2. If you balance the equations, it will be 2, 4, and 4. Are you clear? And this electron will be accepted by reaction center of photosystem 2. And this process takes place at the thylakoid space. Okay? So if you take photo, okay, this is a summary of the entire process. So light energy or photon have been transferred while light harvesting complex pigments, okay, or the accessory pigments to the photosystem 1 or PS1, reaction center complex exciting and electrons of the P700 pair of chlorophyll A molecule. It must be chlorophyll A. Okay, which are located there. So the photo-excited electron is then transferred to PS1's primary electrons acceptor, right? Primary electron acceptor. So creating an electron hole in the P700, which is now known as P700. I purposely put a plus there because lack of electron. Okay. But again, guys, you do not need to produce so detailed when exam. I write for you guys already just now. Those are the things that you include enough already. Are you clear? Okay. So hence creating an electron hole, photoexcited electron are passed in a series of redox reactions from the primary electron acceptor down a series of electron carrier. A series of, it must be electron carrier. It cannot be a series of electron transport chain. Are you clear? Can you see that? A series of electron carrier. And finally, the enzyme NADP reductase catalyze the transfer of electrons from the electron carrier to NADP. Hence, we produce reduced NADP. Okay, so write down the enzyme here, and I don't think I put it enzyme. So enzyme is a NADP reductase. Sorry, NADP reductase catalyze. Okay, sorry. So next, Photons of light strikes one of the pigment molecule in the light harvesting complex again at the PS2, exciting the electrons to a higher level. Okay, so the process continues with your energy being relayed to other pigment molecule until it reaches the P680. Okay, because of the reaction center, P680 is the reaction center of profile A in the PS2. So against this, each photoexcited electron passes from the primary electron acceptor from this PS2 to PS1 through an electron transport chain. It cannot be an electron carrier. It cannot be one electron carrier. Are you clear? Okay, so be careful when you write. Okay? So through an electron transport chain, the components of which are similar to those of electron transport chains that function in the oxidative phosphorylation. So this exergonic means that full with energy. Okay, force of electrons to a lower energy level provides the energy for the synthesis of ATP. So as electron pass through the cytochrome complex, right now to the bracket for this cytochrome complex, again they are electron carrier. Okay, they are electron carrier. So protons are pumped into the thylakoid space, contributing to the proton gradient that is subsequently used in the chemiosmosis, and this electron now pass to PS1. PS1 receive the electron now. Okay. So we have enzyme that catalyze the splitting of the water molecule into 2 electrons, 2 protons, and 1 oxygen atom. Or in general, if you want to put an oxygen molecule, then it becomes 1 oxygen molecule, 4 electrons, and 4 protons. Are you clear? This is 1 atom. 1 oxygen is O2. So double up. So become 4 electrons and 4. Okay, proton. So the electrons are supplied to the P680 pair, replacing the electrons that have been transferred to the primary electron acceptor. So the H plus are released into the thylakoid space. So the oxygen atom immediately combined with an oxygen atom generated by splitting of another water molecule forming oxygen gas. Here, don't worry about the H plus, the proton, okay, released into the thylakoid space and blah, blah, blah. We are going to look at it, right, in detail, in the next lesson, okay? So what is the difference between the cyclic photophosphorylations? If you look at cyclic photophosphorylations compared to the non-cyclic, very, very clear cut here, we know that the electron donated by the same person, received by the same person. Okay, so only involve photo system 1. So look at this. Photo system 1 receive photon, pass the energy to the reaction center, photo excitation of electron take place. This electron will go through the electron transport chain and finally received by the reaction center P700. Basically, I take it out from my left pocket, put it back into my right pocket. Are you clear? So this is cyclic photophosphorylation. But why is it so stupid? When we donate electron and then take back electron. But this process actually allows the plant to synthesize ATP. Are you clear? This process is very important for plants to synthesize ATP. Any formation reduced NADP? No. No formations of the reduced NADP. Any formations of the O2? No formations of the O2. Are you clear? Okay. And what photo system involved for me? Only photo system 1 involved. So if I go back to this diagram now, so basically that is, right? Do you realize it? Instead of going this way, the electron go this way. So when it go this way, continue to produce ATP. Can you see that? Are you clear? Okay. So let us look at the answer. Okay. So first, light energy absorbed by the accessory pigment and then transferred to the PS1 P700. And then electron from P700 is photoexcited and passed to an electron acceptor or primary electron acceptor. And then this electron is passed to an electron transport change or a series of electron carrier. And then this energy is released during the electron flow, and this is used in the chemiosmosis to generate ATP. And last, the electron is then transferred back to PS1. So in the whole process here, we want or plant wants to generate ATP through this cyclic photophosphorylation. Are you clear? So the word photophosphorylation actually is the formation of ATP. Are you clear? Like for example, the oxidative phosphorylation, the substrate link phosphorylation. So this one is the photophosphorylation. It happens during photosynthesis. So let's compare between the cyclic and non-cyclic photophosphorylation. So in terms of photosystem, what are involved in cyclic? Only PS1. Non-cyclic, both PS1 and PS2. Electron flow from the name cyclic, confirm cyclic. But non-cyclics, then non-cyclic lah. Okay. So in terms of the source of electron, who donates the electron in cyclic? So this one comes from P700. And who is the final electron acceptor? P700. Are you clear? So basically from the left pocket, pass it back to the right pocket. Okay. How about the non-cyclic? Source of electron comes from the water molecule. And final electron acceptor is NADP. NADP receives the final electron. So in this case, non-cyclic. Okay, right. Water is the one that donates the electron first, right? The whole process, the whole flow, the net effect. We are looking at the net effect. Net effect actually seems that water is the one that gives out the electrons. So product by the product form for cyclic, only ATP. Non-cyclic photophosphorylations, we have ATP, reduced NADP, and the waste product or byproduct, oxygen gas. Okay, be careful, reduce NADP, not NADP. So with this, I stop for today's class.